Here are all my great slides that I was talking about. So if you have a respiratory disease, it can be progressive and irreversible, and our current treatments don't relieve the symptoms. So often transplantation is the only option. So here is an example of what happens to your lungs if you are impacted by this COVID-19 infection. A serious infection will impact the gas exchange inside your lungs. So here is an alveolus. This is the gas exchange surface of the lungs. And you can see it's lined with type 1 epithelial cells here. This is where gas exchange happens with the blood. And then type 2 epithelial cells here that create surfactant. This surfactant allows this space to expand and contract with low surface energy so that your lungs work. When a person is infected with SARS-CoV-2, what happens is vasodilation increases permeability, and this space starts to fill with a protein-rich fluid. This impacts surfactant secretion, and it also impacts your epithelial cells. And this leads to damage inside the alveolus that can increase inflammation. It can lead to a cytokine response, and eventually the formation of scar tissue. So around here, you'll see fibroblast infiltration, proliferation, and deposition of more proteins. So this part here may result in a chronic lung disease that we might start to see in the future. So here is just one person's prediction. As we see this large increase in COVID-19 cases now, we're thinking that there may be a residual 30 to 50% of these patients that end up with chronic pulmonary fibrosis in the end. And this chronic pulmonary fibrosis is a lung disease that we study a lot in my lab. So now is a really good time to be thinking about how to model lung disease. And it's really important. One of the reasons I believe that we don't have a cure yet is because when we're trying out new drugs, we're discovering new drugs and testing them in vitro, we're often using models that don't quite recapitulate what's happening in a human. So we can learn a lot from growing a monolayer of cells in a petri dish or from running mouse models, but these things don't actually replicate what's happening in a person very well. And so the goal of our lab is to move our models one step further and actually get us closer to reproducing what is happening in a person. But of course, it takes more than cells to make a good lung. So this is a wax casting of what the inside of your lung looks like. It's a very complex architecture. And so scaffolding is incredibly important, but also very difficult to create. And that's because we have a lot of challenges. We have many, many cell types in the lung. The type 1 and type 2 epithelial cells that I mentioned. There are endothelial cells in your capillaries, fibroblasts out around here, and of course, many different cells that make up the bronchioles and the airways. In addition to that, we're working on a very small size scale. So across here, the average diameter is only 200 microns, and across one of your capillaries, it's only 6 microns. So it's challenging to recreate the microenvironment that we find in the lung. Unfortunately, right now, 3D biomaterial models are underutilized in pulmonary research. So I just wanted to show an example. I compared citation frequency to different keywords that I looked up, skin, bone, and lung, in addition to the words 3D biomaterial model. And you can see that over the years, there have been many, many models of skin and bone, but lung down here in pink is still working its way into more use in the laboratory. So we're really excited about that. And our approach to doing this, and the approach of many other laboratories, is to use a tissue-informed engineering strategy. So we do this by relying first on advanced characterization to help us learn more about the lung tissue. We can look at the structure using things like nano and micro CT. We can learn about mechanics through AFM and different types of rheology, composition through mass spec and different proteomic techniques. And then our goal is to use these inputs to inform our design. We can create user needs and characterization, design criteria and specifications, and ultimately a prototype of our model. Eventually, we would like to create a disease phenotype in that model, verify that it matches what we see in a person, and then hopefully translate some of the things we learn back into medical practice. So what have we learned from advanced characterization? The healthy lung microenvironment we're focusing now on the fibroblasts, or the stromal tissue that's supporting the alveolus. It is full of fibroblasts and proteins and proteoglycans. But during disease, this microenvironment changes significantly. And I've just put a few diseases on here that are laboratory studies. So we talked a little bit about idiopathic pulmonary fibrosis. This is fibrosis in the lung, and when this occurs, and the microenvironment surrounding the cells sees a significant increase in organization and cross-linking. This leads to an increase in the modulus, or the stiffness of the surrounding lung tissue, and a decrease in the overall compliance of your lungs. This is often due to an increase in collagen deposition by the cells that are activated. We see very similar types of microenvironmental changes in pulmonary arterial hypertension, which is happening in the arteries in your lungs. And then in COPD, chronic obstructive pulmonary disease, we see kind of the opposite effect. We see a decrease in organization and cross-linking. We see a loss of stiffness and an increase in compliance in the lungs. And so we use these principles to guide how we're designing our biomaterials to mimic the lung microenvironment. So today, I broke our materials down and materials research that's happened in pulmonary so far into 3 broad categories. So we'll talk about naturally derived biomaterials. Most of the really interesting work that has been done in the lung lately has been using naturally derived materials. There's been a little bit of work with fully synthetic biomaterials. And then finally, we'll wrap up talking a little bit about hybrid biomaterials. So hopefully we're getting the best of both worlds when we use these. So we often talk about these design criteria where we're interested in learning about how cell adhesion is influenced in the material microenvironment, how we can control the mechanical properties, and then what's going on with biocompatibility, right? In naturally derived materials, we see that these are very bioactive. They're easily remodeled by the cells. Whereas we have the opposite in fully synthetic biomaterials. We have to add in degradable sequences and like peptide crosslinkers in order for the cells to either interact with these materials or to remodel them. Generally speaking, naturally derived materials on their own don't have excellent mechanical properties, and it's not easy to control these mechanical properties. Whereas fully synthetic biomaterials are precisely tunable. In terms of biocompatibility, I think these can both be tuned to have decent biocompatibility. The naturally derived materials have a higher potential for immunogenicity. And of course, in reproducibility, it's always hard to have very, very consistent naturally derived materials. And so we do see sometimes a batch-to-batch variation there. So I'll just give an introduction to 2 or 3 materials for each category and tell you a little bit about how they've been used to model lung. So for our naturally derived materials, we'll talk a little bit about alginate, matrigel, and decellularized extracellular matrix. So alginate is a naturally derived polymer that comes from seaweed. It gels by cross-linking through ionic interactions, or it can be chemically cross-linked, chemically modified to be cross-linked in other ways. Some of the limitations to this are a little bit of batch-to-batch variation. Actually, the G component here varies from 10 to 70% depending on the season when the seaweed's harvested. And unfortunately, in alginate alone, there are no natural human enzymes that can remodel it. But some researchers have used alginate to do some pretty cool things in lung tissue engineering. So this work done by Wilkinson and colleagues shows that they've created these alginate beads. They've functionalized them with collagen 1 and then put them into a high aspect rotational bioreactor with cells, allowed cells to adhere, which you can see here stained green to show that they're alive with calcium AM. And then they've aggregated these into a petri dish to create a model that looks a little bit like lung tissue. So you can see here, you're starting to see the structure of lung tissue. They've stained vimentin, or the fibroblasts, in green here, collagen 1 in that magenta color, and then of course DAPI is blue. And so using these alginate beads, they've created the geometry that we're expecting to see on lung, and they've done this in a 3-dimensional way. They went on to study how these cells can be activated by exposing them to TGF-beta, one of the growth factors that helps induce the fibrosis cascade. And they've seen increases in both alpha smooth muscle actin and collagen 1 when they treat these systems to recreate fibrosis. Of course, we've all heard of Matrigel, and we all talk a lot about it in the biomaterials community. It is made of a solubilized basement membrane that's extracted from a sarcoma on a mouse. It's decellularized, and then the components you can find in it include laminin, collagen, heparan sulfate, proteoglycans, growth factors. And actually, most of the time, we don't know exactly what's in every single batch of Matrigel, so the composition is not well defined. Of course, there will be batch-to-batch variation. And in general, there's usually fairly poor control over mechanical properties. You can't make a very stiff or realistic tissue out of a Matrigel in general. But in the lung community, they've used Matrigel to make some really interesting models. So this is a model of an alveoli. So here's an image of what the cells look like in a mouse. The white cell staining is the type 1 epithelial cells. The red is the type 2 epithelial cells. And you can see that they're aligned pretty much like you saw in the pictures I showed in the beginning. As these cells grow in Matrigel, we see more and more type 1 cells sort of in the middle here, and then the surfactant producing cells, the type 2 cells, are on the outside. So instead of that really nice monolayer, we're getting buildup of different cell types, but this still remains an interesting model to look at cell-cell interactions that are occurring in the lung. And we think that we can keep tuning models like this in order to achieve a more realistic structure of the lung. Another really great example of work done inside Matrigel is the growing of small lung organoids from epithelial progenitor cells that were derived from the lung in the presence of fibroblasts. And these studies show that when you're growing the lung organoids derived from the epithelial cells, that fibroblast crosstalk influences the size and number of the organoids and also the organization. So we see that when we're putting human lung fibroblasts in an organoid at a certain density, you get a certain size organoid. If you double that density, you get approximately double the size of the organoid. But if you add in these epithelial cells along with the fibroblasts, you see a significant increase in the organoid size. So these cells are communicating with each other and changing the size and structure of the organoid. So if you look at the fibroblasts on their own, you see a large cluster of cells with no hollow interior. But if you start to add the epithelial cells, you start to coax these cells into growing into a structure that looks more like an alveolus with an airspace in it. Similarly, these cells, when they're crosstalking with each other, impact activation or that fibrotic response of the fibroblasts. So if you co-culture fibroblasts with epithelial cells and then expose them to something like TGF-beta to induce fibrotic activation, in the presence of the epithelial cells, the fibrotic activation is generally lower than it is without them. So these have been really interesting studies that have been performed in Matrigel materials. Decellularization is a really hot topic in the lung field, and it has been worked on a lot because there's a hypothesis that if we're able to decellularize lungs that were not originally suitable for transplant and then recellularize them, that we might be able to replace lungs and transplant lungs that were not suitable for transplantation to begin with. So when we take the cells out of lung tissue, it looks like this. This is a picture courtesy of our collaborator, Darcy Wagner, at Lund University in Sweden. And the composition is very similar to that of Matrigel. It is composed of collagens, laminins, elastins, cell adhesive proteins, and growth factors. There have been some studies showing that proteoglycans and glycosaminoglycans might be damaged in the decellularization process. And so that's ongoing to see if we can replace those and then recellularize the tissue in the way that it was before. Again, some limitations to this are what we would expect. Some batch-to-batch variation depending on where you get the tissue that you're decellularizing. Some lungs are healthier than others. Some lung sources are more consistent than others. And of course, control over mechanical properties is also difficult when using this decellularized extracellular matrix to create a hydrogel biomaterial. Here are some images of what it looks like when we decellularize lung tissue. So the native lung tissue is on this side, and then each row is labeled for a different component. So you can see once decellularization happens, the nuclei that are stained blue for DAPI disappear, and you can see that left over you have things like fibronectin, laminin, collagens 1 and 4, and of course elastin, which helps make your lungs stretchy. It's then possible, once you've decellularized these constructs, to recellularize them, and this has been done using human lung fibroblasts, basal epithelial cells, mesenchymal stem cells, and also cells from the bronchioles. And so it's been very interesting to watch this field grow. We've actually seen that you can recellularize a lung and put it back into a mouse or a rat and maintain gas exchange for up to 6 hours. So this is really promising. Something similar has been done in a larger model, in a porcine model, but the amount of time gas exchange was maintained was pretty short. It was under a few hours. So it's still a work in progress, but this is a field that is growing and working hard to see if we can create lung transplantation alternatives. As I mentioned previously, you can also use decellularized lung tissue as a biomaterial all by itself. And so a recent study, which was really fascinating, showed us that if you use these decellularized constructs, here it is a trachea and then a lung, and compare them to our typical in vitro models, growing organoids in 3D and matrigel, or differentiating airway cells at an air-liquid interface, you can see that the tissues that have been decellularized create location-specific regeneration of cells that are put on these tissues. So an engineered trachea or a decellularized trachea will result, based on RNA-seq analysis, in more mature proximal epithelial cells than you would find in an organoid culture or in an air-liquid interface culture. And the cells that were put inside engineered lung tissue actually start to produce surfactant and start to differentiate into the alveolar type 2 cells that we saw previously. So it's really fascinating that the proteins and the structures and the microenvironment that's left over when you decellularize these tissues is so instructive that you begin to get location-specific regeneration. If you take these materials that have been decellularized, lyophilize them, crush them up into a powder, and then turn them into hydrogel biomaterials, you can see that decellularized extracellular matrix, or DECM hydrogels, resemble healthy and diseased lung tissue mechanics. So this is a study out of Jeanette Burgess's group, and they're showing Young's modulus here, and then in red is the tissue that they tested, and in blue is the hydrogel made out of the decellularized tissue. So you can see control healthy lungs, they sort of range from 1 to 5 kilopascals in Young's modulus, and when you recreate that with a hydrogel, you get something similar, a little bit lower modulus. In COPD, you have a slight drop in modulus from your healthy control samples. And then in IPF, please notice that this scale is much different. So this is 40 kilopascals instead of 6. And you see a significant increase in the modulus of the tissue that has started to be replicated in the modulus of the hydrogel. So it's really fascinating that these hydrogel materials also retain some of the mechanical properties that have been changed in the microenvironment of lung disease when the lungs are no longer healthy. Other research groups have taken these types of materials and actually grown cells in them or on them. And so you can see here that these hydrogel biomaterials that are made out of DECM support cell viability, and they've also been able to support mesenchymal stem cell delivery to the lungs. So these MSCs were encapsulated in the hydrogel biomaterial and delivered to specific places inside a mouse lung. So that wraps up our naturally derived biomaterials. It's been really exciting to see the field grow and to learn more about it as I've entered the field of pulmonary engineering. I spent most of my life studying fully synthetic materials and arguing a lot that they were way better than natural materials. So it's been fascinating to learn all of the really amazing things that have happened with fully natural materials and to start to incorporate that into my thinking when I think about how to model lung disease and regeneration. So in the field of pulmonary engineering, as I mentioned, our 3D biomaterial models that we see a lot at Society for Biomaterials conferences are not used quite as often. So I'll start out by telling you about two materials that seem pretty old and kind of like they have been used a lot in settings where we were just starting to implement biomaterials engineering to study diseases. And so we'll talk a little bit about polyacrylamide, which of course the biologists that are interested in studying cellular responses in the pulmonary field are very familiar with because it's been used for ages to run gels. Polydimethylsiloxane, PDMS or silicone, and then of course my favorite material, polyethylene glycol. So polyacrylamide is a fully synthetic polymer that we can cross-link to create a hydrogel that's hydrophilic and swells with water. It has a tunable stiffness. We can tune it anywhere from 1 to about 70 kilopascals. And just like other fully synthetic materials, it inherently does not have any bioactivity. It requires the addition of cell adhesive ligands or coatings, and it's not easily remodeled by cells. So again, it requires the addition of biodegradable sequences if you're interested in having it be remodeled. But polyacrylamide has shown us some really interesting things in the lung. So in 2010, this work from Dan Schumperlein's group showed us that if we create a stiffness gradient in a polyacrylamide gel from 0.1 kilopascals up above 50 kilopascals, you can see that we get a gradient of activation of these human lung fibroblasts when we grow them on the materials. They've also gone on to show that there are changes in migration speed and persistence based on the stiffness of the underlying matrix. And they've shown differences between healthy saline and bleomycin. This is a drug that is administered to mice to create a fibrotic lung response. They've seen differences in those cells on these different biomaterials. So it's been really fascinating to see this work emerging in the pulmonary field and to see this group building on it. So here they have used pulmonary artery smooth muscle cells, and they've shown that YAP-TAZ activity is required for stiffness-dependent remodeling in these cells, and they've done this again using a polyacrylamide hydrogel base. They've also shown that cells that came from a pulmonary arterial hypertension or PAH patient are inherently more active on these surfaces, whether they're soft or stiff, than the control cells. So you can see in the PAH cells that you have nuclear YAP here, even though it's being grown on a soft material. All right, silicones. I spent most of my early career studying anti-fouling, and we used silicones to keep things from sticking to the sides of ships. So this is a fully synthetic cross-linked polymer. It makes a hydrophobic network. It's often filled with silica and platinum catalyzed. This is, again, a tunable stiffness substrate. You can create silicones that have a modulus of 5 to over 100 kilopascals, and it has the same types of limitations as the previous materials that I mentioned before. One of the newer studies that I've seen out of the pulmonary field that has been really fascinating is this. They've used PDMS micropillars to enable the assembly of these tiny lung microtissues. So they created these pillar arrangements based on the size and shape of the human alveolus, and then grew fibroblasts, thin layers of fibroblasts on top of these. These cells were then treated with TGF-beta to induce fibrotic activity, and you can see an increase in alpha smooth muscle actin and procollagen, and of course fibronectin. And then you can also see that there's an increase in the contractility of these cells on these surfaces. So it's been a really fascinating way to create a thin layer of tissue that starts to mimic lung tissue, and to look at how these cells are responding when they are challenged with something that would be profibrotic. Another demonstration of PDMS being used in pulmonary engineering has shown us that mechanical memory in lung exists in lung myofibroblasts. So these are fibroblasts that they've grown on different stiffnesses of materials, and if they start out growing them on a very stiff material, 100 kilopascals, and then put them on either a stiff or soft material, they see that this mechanical memory, this activation in the cells where you see enhanced alpha smooth muscle actin expression and organization, is persistent even when these cells are plated onto a soft material later. So it's been a very powerful way to study how these cells are responding and how we could potentially reverse disease. Obviously, it's complicated because of this mechanical memory in the cells. It also reminds us that if we're growing our cells on tissue culture polystyrene or regular Petri dishes, that it's important to think about how we have pre-programmed our cells before we test them in our own biomaterials. Of course, my favorite material, I did my postdoc in Christy Anseth's group, and so I spent a lot of time looking at and learning about polyethylene glycol. This is, again, a fully synthetic polymer. It can create a cross-linked hydrophilic network, and it has a tunable stiffness somewhere between 1 and 50 kilopascals, not quite as stiff as PDMS. In our lab, we use PEG norbornene quite a bit and a few other PEG-based polymers. This is just an example of a PEG network where we can put in PEG norbornene multi-arms and then cross-link it with a dithiol PEG crosslinker and add in binding moieties like RGD and YIGSR that mimic fibronectin and laminin inside the body. One of the really cool studies that has used PEG to look at lung structure came out of the Anseth research group. So they created photodegradable PEG hydrogels that were in the shape of microspheres. You can see they were here. They've surrounded them with epithelial cells, and then once you shine light on these materials, the PEG inside the alveolar-like structure goes away, it degrades, and you're left with something that looks kind of like a lung. This is really exciting. They're showing here labeling T1 alpha is those type 1 epithelial cells, and surfactant protein C are the type 2 cells. So they're starting to get a mix that is a similar ratio to what you see in your lungs. And they've used this model to study how fibroblasts migrate around these alveolar type geometries. So the alveolar system is here, surrounded by green fibroblasts here. And you can see there are differences in migration if you use a regular healthy epithelial cell versus an A549 epithelial cell that was derived from a cancerous site in the lungs. So this has been a really fascinating model to look at crosstalk again between these two cell types, but now in 3D in a system where we can finely tune the material properties and really control what the cells are being exposed to. In our laboratory, we have created a polyethylene glycol embedding matrix that helps support precision cut lung slices grown ex vivo. So precision cut lung slices are very, very thin slices of lung tissue that can be grown ex vivo and studied in a way that maintains the architecture and the multicellular microenvironment inside the lungs. Unfortunately, these precision cut lung slices on their own don't last very long ex vivo. The controls on average are only able to be kept for about 7 days. That's a huge limitation when you're studying chronic pulmonary diseases because you need more time, obviously. So our laboratory has embedded these in a PEG norbornene-based biomaterial. So our precision cut lung slice is inside the biomaterial. This is an image of a slice through the middle of this. So ecadherin labels the epithelial cells and then DAPI to show you the nuclei. And basically we used a design of experiments approach to optimize viability, surfactant protein C production, so functionality of the AT2 cells, and then to decrease vimentin or to reduce the number of fibroblasts that are proliferating inside these materials. And we landed on a material that contains specific amounts of a specific thickness and specific amounts of RGD and YIGSR. And this has helped us to extend ex vivo culture of our precision cut lung slices from 7 days up to 21. One of the most exciting things that's come out of using fully synthetic materials to model the lung, it came out of Jordan Miller's lab. So they have been able to 3D print this incredible system that mimics an alveolus and the capillary network around it. And so you can see here, there's an alveolar structure. It can be filled with air. The capillaries around it can be filled with blood, and they can actually do gas exchange and oxygenate the blood inside this 3D printed model. So this was incredibly exciting. And when I was talking to my MD colleagues in the pulmonary division, it blew their minds. They had no idea that we could engineer something this complex. So it's just a testament that even though it is very challenging for us to model the 3D microenvironment of the lung, we are, as a field, we are moving in that direction. And I think it's an incredible time to be studying pulmonary engineering. So I'd like to finish up today by talking a little bit about hybrid biomaterials. These are biomaterials that are some combination of a fully synthetic material and a naturally derived component. In general, I'll be talking today about cross-linked hydrophilic networks that have a tunable stiffness somewhere between 1 and 50 kilopascals. And I think there are some advantages here. So, and we have inherent bioactivity that is provided by the naturally derived component. The cells are typically able to remodel that naturally derived component. And because we have the synthetic component involved, there's better control over mechanical properties. So one recent example of this came out of April Clarkson's lab, where they've used hierarchically structured hydrogels for 3D cell culture. And what they've done here, their natural component is a multifunctional collagen mimetic peptide that forms a triple helix fiber assembly. And they've been able to use that to cross-link their polyethylene glycol component to create a 3D hydrogel network that contains both natural and synthetic components. And you can see inside these networks that the cells are alive and thriving, and there is actually a dose-dependent response of cell morphology and spreading to the collagen peptides that are inside the material. So you see here, the larger dose of collagen peptides induces more spreading and elongation of the cells versus no collagen peptide inside the material. So I think this is an exciting way to start studying some of the things that are going on in a collagen-rich microenvironment, such as a lung that has experienced pulmonary fibrosis. Recently in our lab, a master's student, Cassandra, just published this paper. It's in the Journal of Materials Chemistry B. And what we've done is used a PEG alpha methacrylate. So this PEG material, the methacrylate is attached sort of on the opposite side as you would usually. And when this ester bond cleaves, an alcohol is released, but the PEG backbone stays intact. So we've created a PEG methacrylate hydrogel that has higher hydrolytic stability. And then we've cross-linked this with a thiolated decellularized extracellular matrix material. And we've been able to tune our materials so that we could create a dual stage polymerization where our first material is cross-linked in a Michael addition off stoichiometry to create a soft material. And then we add a photoinitiator, initiate a homopolymerization of the remaining methacrylates, and create a stiffening event that stiffens our material into the pathological range. So you can see from our confocal images that if we label the PEG and the DCM, we get a good mixture of both components. It's been really exciting to work on these types of materials because we have both temporal control. We can stiffen over time and achieve activation of these dual transgenic reporter mouse cells. So we see expression of collagen 1 and alpha smooth muscle actin increase in our stiff and stiffened microenvironments. And we can also use light to control where we stiffen. So we have spatial control over stiffening that will allow us to start to recapitulate the pulmonary fibrosis microenvironment, where the changes in lung stiffness are not homogeneous, they're heterogeneous, and they change over time. So our mission, and the mission of many other labs out there, is to design biomaterial-based models of lung disease and regeneration, and hopefully we can use these to improve our success rates for identifying new drugs that we can use to treat patients, especially at a time like this when we anticipate there will be a significant increase in chronic pulmonary diseases that still have no cure. So I just wanted to thank Society for Biomaterials, Ashley and Dan and Sheena, and the Engineering Cells and Their Microenvironments, SIG. Of course, my laboratory has been instrumental in all of this work, as well as our collaborators and funding sources. So thank you for your time.
Okay, that was fantastic. Thank you so much, Chelsea. So we'll now open the floor up for questions. And as a reminder, if you have a question, please type them into the question box. I currently don't see any questions. Dan, are you seeing any that
I am not either.
Okay. Just making sure I'm not missing anything. Well, while we give some folks time to ask questions in the question box, I have a few questions related to your intro. So you discussed how the work that you're doing relates to COVID, which of course is on everyone's mind right now. And you talked about the likelihood that fibrosis is going to be an issue in these patients moving forward. Do you know off the top of your head what the incidence of fibrosis was in SARS survivors?
Oh, that's a really good question. No, I don't. The only thing I can think about, there's sort of two ways that fibrosis could happen, right? First, it could happen because of a very severe infection, but also it could happen if you end up on a ventilator. And so I think there will be differences in ventilated versus non-ventilated patients, and the ventilated patients will probably have a higher rate of fibrosis.
I see. So in ventilated patients in general, do you know the incidence of fibrosis?
I don't know the number off the top of my head. No, but it does occur, and so one of my colleagues at the university studies that quite a bit.
Great. And your stiffening materials are just absolutely fascinating. What is the time scale of that stiffening, and does it maintain over time?
Yeah, that's a great question. Right now, the time scale is pretty instantaneous. So we're initiating a radical reaction. It happens within seconds. It would be fantastic if we could titrate that to be slower over time. So it's something we're thinking about. Once the stiffening reaction has occurred, the material remains stiff.
Okay, indefinitely. You've looked at that for a while.
We haven't measured it, but theoretically it will. So we've looked at it for several days, but not for months yet. Oh, actually, never mind. We have. We did hydrolytic stability studies where we stiffened our materials and looked at them for 60 days, and they maintained the stiffened modulus values.
Wow, that's fantastic. And I'm assuming in these in your materials, it's the decellularized ECM components that are providing. And I apologize if I missed it, but the decellularized ECM components, is that what's actually providing attachment sites for cells that you're incorporating?
That's a good question. We believe it is, but because we were comparing a lot of things to a synthetic control, we ended up putting RGD in both of these materials. And one of the next steps for our lab is to start to explore these decellularized ECM hybrids without the RGD added.
I see. Fantastic. I'm still not seeing any questions in the pane. Dan, just checking in. You're not seeing any either.
I am now seeing several questions.
Okay. For some reason, they're not showing up in my chat window.
They're in the questions pane, not in the chat pane.
Right. I'm looking in the questions pane as well, and they're not showing up there for some reason. Maybe you could.
Sure. I'll just begin to run through them if that's okay.
Sure. Thank you.
First one, great talk, Chelsea. What are your thoughts on incorporating immune cells in these models?
That's a fantastic question. We have to do it. They're so important in all of these diseases. We are currently working with a collaborator to see if we can incorporate immune cells into our precision cut lung slices that are embedded in hydrogel materials. In general, I think we'll have to have a loosely cross-linked network so that these cells can travel through the hydrogel materials. But I think it'll be possible. We'll have to control for the impact of the material on the immune cells as well.
Thank you. Next question. Can you comment on decellularized versus devitalized matrix that you've seen or used for lung tissue engineering?
I guess I'm not familiar with the term devitalized.
Okay.
So my experience is that I've only used fully synthetic and then fully decellularized extracellular matrix. I haven't ventured into myself using very many of the naturally derived materials yet.
Okay, next up, hello, this is Uday from University of Texas at Arlington. My question for today is, what is the role of stem cells in remodeling the lung during injury or fibrosis? Can we revert fibrosis completely, or to what extent?
So that is an ongoing debate in the lung community. There are folks that believe there are cells in the lung, progenitor cells, that can regenerate small amounts of lung tissue. That's one side of the argument. The other side is, no way, we're not going to be able to regenerate lung based on what's already in there. So the AT2 cells that I mentioned are progenitors for the AT1 cells. And when there's a small amount of damage to the AT1 cells, the AT2 cells will dedifferentiate and regenerate that area. I don't think that this can happen on a very large scale. And I think we're still doing a lot of work to understand if fibrosis is reversible. There are some laboratories that are trying to reverse the cross-linking of collagen that occurs and use different drugs to address these things. I think we don't know the answer yet.
Thank you. Next question. Have you done any drug screening experiments within which there was a difference in vitro and ex vivo model?
I haven't done any myself, but we are gearing up to start working on some in these precision cut lung slices. And I think it'll be fascinating to compare the ex vivo and the in vitro models. So it's a great idea. Haven't done it yet.
Next question, for the decellularized lung matrix hydrogel comparisons with tissue, does the fact that the hydrogel trends with disease state and stiffness pose a problem for use in the clinic and hopes for implantation?
Well, I think if we were planning to use decellularized extracellular matrix to heal a patient in the clinic, we would want to use healthy, well, I would say relatively healthy lung to create the decellularized extracellular matrix instead of diseased lung. So hopefully that doesn't become a concern.
Next up, a chicken and egg question for PAH. Do you know if there is first a cellular change or a matrix change at the beginning of the disease?
That's a great question. I debate about that all the time. And also, there are multiple cell types. I only mentioned fibroblasts and smooth muscle cells today, but there are multiple cell types. So the cells could be changing first and then secreting matrix that then changes more cells, or there could be changes in flow and changes in pressure that influence the matrix, cause the matrix to stiffen, and then cause the cells to activate. So I don't know the answer to the chicken and egg question yet. It's a good one. It's fun to talk about.
And the last question I have in my queue. Hi, thanks for the great talk. Have you or anyone performed the mechanical testing in a media that represents the actual lung? For example, fatigue testing in a specific media and simulated movements of lung?
Oh, that's an excellent question. No, I haven't done that myself. The paper that I referenced that was showing differences between tissue and the hydrogel biomaterials, they did some stress relaxation and some more complicated mechanical testing. Also, they weren't trying to inflate the biomaterials and deflate them and fatigue them like a lung. But definitely check out that paper, because I think you'll find some more interesting mechanical testing in there that is related.
Thank you very much. And thank you, Chelsea, for a great talk. Thank you, Ashley, for hosting today. Before we go, just a couple of quick housekeeping announcements. Please be aware that we're working to postpone the fall symposium in Hawaii. We will not be there in December, but likely in late July 2021. In addition, in the coming weeks, we'll be issuing a call for ideas for the 21 annual meeting in Chicago. That'll be April 21 to 24, 2021. And pardon me. We are also working to reschedule the SIG social and the Young Scientist social. to be held during the World Biomaterials Congress, now scheduled for December 11 to 16, 2020 in Glasgow. And yes, I'm still planning to organize a distillery tour. All members are being asked to participate in a needs assessment survey. As we adjust to the new normal, we'll be exploring ideas for new member benefits, and we really value your input. Please respond to the two-part survey. The deadline for part 1 is today, so you should have received emails on that, and you should receive an invitation to complete part 2 tomorrow. And lastly, please be on the lookout for an announcement regarding the scheduling of an SFB members-only annual business meeting. This meeting is an important transition for SFB as it represents a changeover in leadership terms, with Horst von Rechem passing the presidential gavel to Shelley Sekiyama Elbert. Please stay tuned for more details on that in the near future. Thank you again for your participation today, and please reach out to me at info@biomaterials.org if we may be of any service whatsoever. Thanks again, Chelsea. Thanks, Ashley.
Thank you. Thanks, Chelsea.